Well, welcome everybody to Stars, Cells, and God. This is the show where we discuss new discoveries taking place at the frontiers of science that have theological and philosophical implications. These discoveries also have implications for the reality of God's existence and the reliability of Scripture. My name is Rana. I am the Vice President of Research and Apologetics at Reasons to Believe, and I am your host for today's program. And today we're going to be talking about the grand plan to track diversity in science journals, as well as the human genome sequence. But before we get into today's discussion, I wanted to encourage everyone to subscribe to our YouTube channel, Reasons to Believe, and click on the bell icon so that you can be informed of our new videos. We release new episodes of Stars, Cells, and God every Thursday. Also, go to our website at reasons.org, which is full of excellent resources on a variety of science faith topics. To learn more about Reasons to Believe, you can also follow us on social media at RTB_official. I'm joined today by Dr. Jeff Zweerink, a good friend of mine, who is going to open us up today with an interesting discussion about diversity and publications in science journals. Jeff.
Well, thanks, Fuzz. I appreciate the time to be here, and I'm going to say straight up front that I'm a little terrified to have this discussion, just because I think there's so many ways to misunderstand, kind of what I'm saying, and really what I'm raising, just straight off the bat, I'll say is this question of, given the way we talk about race and all of this, race, diversity, discrimination, all of that, the way we talk about it, there are aspects of that the way it plays out in popular culture, popular society, seems at odds with the way science works. And I just want to have a discussion about that. I'm not out here advocating that we do X, Y, or Z, but I think if you don't have the conversation, then you're not clear what you're talking about, and you can't actually make real progress on solving the fundamental problems. Because I do think that there are some very clear data of what the situation is in academia, but then there's also conflated with what are the issues. And one of the things I love about science, and I say this in classes where I teach, is that in science, one of the cool things is that anybody can bring their ideas to the table. And the beautiful thing about science is not that your idea doesn't get accepted because you're prestigious. It doesn't get accepted because you're oppressed. It doesn't get accepted because you have the most people behind it. In the short term, those sorts of things may play out, but ultimately what validates your idea and acceptance is, does it line up with the world? Because you go out in science, you say, here's my idea, let's go out and test it. What sort of measure could I do, or what sort of data could I take that would weigh in on this? And if the data doesn't line up, you go back to the drawing board and you either say, hey, my idea is wrong, or I need to modify it to account for the new data and then retest it. And so the beauty about that is that my ideas and what I think are not, hey, how well can I put them together? It's, can I put them together in a way that aligns with this external standard? Right. And I think that's why science has accomplished as much as it did. I think ultimately science was developed just to try and understand the world, but why it's accomplished so much is that it is willing to say, all right, this idea doesn't line up, this idea, this model, this way of looking at things doesn't line up the world. Let's modify it or scrap it and come up with a better way of looking at it. And this repeated testing, because you'll find throughout the scientific literature ideas that seemed really elegant, really good, they just seem to make sense, and then you go out and you make the tests and they don't hold water. And so as I was just reading through various scientific journals, I came across this article, and it says, a giant plan to track diversity in research journals. And, for whatever reason, as I read through the title, the thing that caught my mind is, wow, that's an interesting juxtaposition of kind of how we treat discussions on race and gender and class and all that in society, and the dynamics of how science works. And what I don't want to get missed in this discussion is that there's data that gives a picture of what the community looks like and how things happen. But then, if we are not careful, we can miss how science addresses those problems. It goes in and says, all right, how do we best explain that? And only once we best explain it, then do we step back and say, all right, what are the mechanisms for moving forward? So, in a lab, I can go and say, all right, I got this problem. I can start throwing ideas at it, or I can say, okay, well, this is the way to solve it and go at it. Or I can say, hey, let me get a good understanding of what's going on. And when I do that, I'm far more likely to get to a good solution to my problem quickly.
Right.
And so that's kind of really the nature of what I want, the discussion I want to have.
Sure.
Because, if you can, one of the things that as people have talked about this is there's a disparity in how many journals are published by what people, how many people of different genders or different ethnicities are in different positions. And one of the things that people found is that there's just a lack of data on what it is out there because there's nothing in the scientific journal that identifies outside of your name that it identifies much about that because the history has been, this is my name, this is who I work for, this is what we're talking about. And so that whole discussion of are you male, female, or other, or are you black, white, whatever, that just hasn't been part of the discussion. So there's been no data collected. And so there's kind of a push to get more data collected on that. And I think it's great. You can kind of go to the first slide.
Sure. Some
of the things that have shown up, and again, I'm going to identify some of the challenges in getting really good data. But, just looking at this is the family of authors and reviewers at AAAS, a very prominent scientific society that, 20% of the people, you got to realize that most of it has no data. You're up to almost 70% there that has no data. But of the remaining 30%, 20% is white, 10% is Asian or Pacific Islander, very small fraction of Hispanic, Black, African, American Indian, Native American. And that sort of trend or that sort of distribution kind of goes, the ACS has the same thing. You look at editors at high-profile journals like Science, again, the same sort of phenomena, Journal of the American Chemical Society, Journal of the American Medical Association. What you see is this compared to the distribution of ethnicities in society or race in society, there's this disproportionate amount of white people in those, both as authors and as editors and people who have authority and positions. And so that's the data. I mean, there's just no way around that. That's what the data says.
Now, does this data correct for or take into account the occurrence of these different racial and ethnic groups within the population, right? So like if you say in the United States, I don't know what proportion of our population are white versus African American or Hispanic, but you would expect to see if everything is an equal and even playing field, then you'd expect to see that the ratio of the different types of people that are publishing in a science journal would more or less correspond to the population, right? So
yes,
because in a sense that these papers are drawing authorship from a global audience. That's the
idea.
Yeah. So
I don't know the specific answer to that. I know one of the things that is true is that it's very difficult to get this data. And so the way some of this data is generated is, you read through the article and sometimes people will just go and scour Wikipedia pages and get information on who it is. And so that's how they're compiling the data. Or they will use AI name recognition where they're looking at names and identifying names associated with certain ethnicities or races. And so, you can ask the question, statistically, does this work or not? For an individual person, you can very easily be wrong, but your question is, statistically, does it work? And you raise a or at least one of the things that I heard in there was another important point, is that it's important that I don't think it's appropriate to compare this to the average population in the United States, because really what you're looking for is bias in publication, which is reflective of the population of scientists. So it may be that we've really biased the scientists, but depending on, once you get into the scientific community, your papers get published at the same rate as everybody else. And so these are all the challenges that arise in doing this sort of analysis that are amplified by the fact that it's really hard to get the data in the first place.
Right. But I think you and I would both agree that generating this kind of data is really important, right? Because we want to understand, is there some kind of systemic, racism or, systemic bias, maybe is a better way of saying it, within science that is maybe, precluding certain people because of their ethnicity or their gender from actually participating in science.
Right.
So that the data is really important, I think, to understand, right?
No, it is, and thus raises some of the tensions in how you go about doing this, because one of the best ways to get this data is to just have people identify. You ask the question, you get the data when it comes in. One of the problems you have to recognize with that is historically, you go back into the 1930s and 40s, and that was the mechanism that people used to collect data on Jews in Germany, and that was used to systematically persecute, and that's what led to the Holocaust. And so this isn't just everybody's going to use this. Well, this is pretty sensitive data that could be easily misused. Right. And so, there's a whole lot of challenges just in how you go about doing this. I mean, and you've highlighted that one. There's another issue of, you mentioned this is worldwide, so anybody across the world can publish in the APS, Journal of American Chemical Society, whatever journal. There is no, you got to be in this country. Well, different countries delineate these categories differently. And so even, so now you have to systematize across the world, which is really quite a challenge. And inevitably, you run into different worldviews there. Not going to spend a whole lot of time, but in the West has a more Judeo-Christian history to it. There is very much this idea that all people are created equal and that they have value, and so that really is one of the foundations, I think, of why science developed and flourished in the West, was because of that idea. Again, don't read more into this what I'm saying, but if you've got the caste system in India, there are certain people who you don't want to publish because they're undesirable or whatever. And so now you've got to deal not only with different ways of categorization, but there's different worldviews in play there. And so it's just a really complicated, in some sense, it's a really fun and challenging scientific problem, but it's not all we have to do is X, Y, or Z and we're going to get it.
Well, and if there is a journal or journals that are being published in China in Chinese, you're going to see obviously a skew in the demographics because of language inaccessibility. It doesn't mean that intrinsically that journal is systematically biased against people of different ethnicities, but if you don't write Chinese, you're not going to be able to publish in that journal. So that seems to me to be another complication.
No, and that is one of the big complications, and this is where I think we want to have a really good, robust discussion on what is it that we're trying to do here, because like I said, there are these inequities in terms of how it's representative of the population in general. Where in the chain that is, that's a whole different area of research. But one of the ways that science works well is to say, okay, this is what the data is. How do I go about explaining that? And one of the things that I found interesting was that there were studies that show that even when you account for the fact that there are fewer minorities in science, that minority-based publications tend to have fewer citation rates, and so there is another bias that is going on there. And so this was, somebody who'd done that study, said at a 2020 preprint from a team led by physicist Daniel Bassert at the University of Pennsylvania, found that authors of color in 5 neuroscience journals are undercited relative to their representation. Okay. The team's analysis suggests that this is because white authors preferentially cite other white authors. Now, I don't dispute that white authors preferentially cite white authors, but they make it sound like it's a race-related thing. My suspicion is that is I cite people who I know. I know you've done work interviewing for jobs, and you get thousands of applicants. How do you go through and sort through thousands of applicants? Well, I do know this. When I'm looking through something and I know this person who I know that person, and they say, it just carries more weight because there's more data behind it. There's more trust. There's more there. Right. And so I'm going to preferentially cite the people that I know.
Right.
Now, if I happen to know other white people, that's who I'm going to cite. And so I think this is an argument that we need to get a broader group of people into the community. But that problem may well solve itself after that, because my suspicion is people of color are going to cite people of color if they know them. And it's not a race thing. It's something else, but it could very easily get categorized as a race problem.
Right. Well, you're bringing up a really interesting point, because if you think about the idea that, again, science is supposed to be strictly objective, where it doesn't matter who that author list is. On the paper, when people are peer reviewing it, it's, is this actually a sound paper, a scientifically sound paper? Are the conclusions that are being drawn, sound relative to the data? Have the experiments been performed well, right? Those types of things. So really, to your point, and I'm highly sympathetic to it, is that if we can actually improve the diversity of people entering into the sciences, the problem should actually resolve itself in terms of any kind of bias in the publication or in the citation rates, right?
Well, yeah. So this is where I think the way science approaches things, and again, it's that what does the data say, what are the tests, what do the experiments say, is somewhat in conflict with the way we deal with things in general conversations about things, and it's. I'm not sure what we do with that, because it has this flavor, and I'll show another article, and this is just an example of what I'm concerned about. If you can show the next slide there. So this was, people have been talking about the structural racism, and so there was a discussion between Dr. Ed Livingston and Dr. Michael Katz that they had a discussion on structural racism, and Dr. Livingston apparently somewhere in this podcast made a statement of how can structural racism exist because racism is illegal. And it created quite a stink. And you read there, this has been, it's been withdrawn. Please listen to the apology from this other doctor who was his supervisor. And if you read down there at the bottom, it says, has been withdrawn, made in because comments were inaccurate, offensive, hurtful, and inconsistent with the standards of JAMA. Racism and structural racism exist in the U.S. and in healthcare. After careful consideration, determined that the harms caused by the privilege outweighed any reason for the podcast to remain available. That doesn't strike me as the way science works, because there's this data out there of what he said. I cannot listen to and evaluate the ideas because it's now been hidden. Again, I look through the history of scientific ideas. There have been controversial, challenging, provocative ideas that have been put there, and when they're wrong, there may be an errata produced or something that says, hey, this was incorrect, we're going to withdraw this paper, but it's still there for people to see because it's data that needs to be incorporated and talked to. And there's a whole lot to this situation that I don't know, but it seems like what's going on here is, hey, this hurt people, but the data is still the data. Right. And as a scientist, you don't get to throw out data because it doesn't align with X, Y, or Z. You have to explain it, account for it, or say, well, we're going to put it over here until we have more data or something like that. And so this is kind of that tension that I think we need to just have a discussion of what do we do with this? Does this mean that well thought out but provocative ideas can they be ruled out just because of reasons that are kind of unscientific, if you will? This is the question I have, because that will ultimately undermine the fabric of science and the reason why it works so well.
Well, your point reminds me of something I oftentimes see in apologetics that I try to avoid. It may be with marginal success, but when it comes to I'm a skeptic of aspects of the evolutionary paradigm. And part of my concern with ideas like human evolution are, what are the implications of this idea, right? But I see a lot of apologists highlight the implications of human evolution, and then on that basis make the argument that this is really an idea that we need to reject, right? Now, to me, just because I don't like the implications doesn't mean the idea isn't true.
Right.
Right. Now, if I don't like the implications of the idea, that to me sets up red flags where I ask the question, how am I really certain that this idea is true? Or are we really sure this idea is true? And so I'm going to investigate the idea with a greater degree of scrutiny than I might ideas where I'm not as troubled by the implications. Right. Right. And I think that's, in my mind, that's legitimate, at least, to say, hey, I don't like the implications. I really want to make sure that these claims are sound before I accept them. But again, what I prefer to be true doesn't really make something true. And it seems to me like there's an analogy to what's happening here is, I don't like what you've said. And maybe what you said is actually hurtful to some people, right? That is that these are the implications, but is what you said true or not?
Right, yeah.
And how do we know if it is true?
Right. Well, and I think that's where, there's, Ken Samples has made the statement that, for a long time people were asking truth claims or asking truth questions. And I think that still goes on, but they're asking, is Christianity good? Well, this is one place where I think the worldview that Christianity brings gives us a great framework for dealing with these sorts of problems, because there is this inherent property of Christianity that you're pursuing the truth. And I think, and one of the aspects of pursuing the truth is that God is who he says he is. Christ has redeemed us. God cares about people. We're supposed to go out and love others the way God loved us. And I think a lot of the motivation behind the diversity, equity stuff is to, hey, we're not loving people well. There's these inequities. We want to correct them. There's that drive in there. But we need to, there's with that drive of wanting to care for people, we need to maintain that emphasis on the truth, that both of those can work well. And the solutions that work are the ones that care for people and maintain the truth.
Right.
Not all worldviews share that way of looking at things. There are worldviews where truth is irrelevant. There are worldviews where caring for other people is irrelevant. You want a worldview to be correct that has truth and caring for people both being a fundamental aspect. And whatever other worldviews have to say, I'm not expert in those, but I do know Christianity is driven by the truth as well as caring for others.
Yeah. Well, I think the value in these kinds of publications is at least calling attention to the problem. But we just want to make sure that when we call attention to the problem, that as you said, I think really, really well, is that we really put forth genuine effort to truly understand what is undergirding the problem so that we can figure out what is the best way to draw the solutions. I think that's a really well-made point.
Yeah. And really, a lot of my thinking about this is that, obviously, I wouldn't be on this podcast if I weren't passionate about Christianity, but I just see a number of ways where the integrity of science is being undermined in the public's eyes. We could have long discussions on how the pandemic was handled, but I think some of the ways that done undermined people's respect for what science had to say, not because science didn't have this way of doing things, but because there's this perception that science becomes biased or whatever. Right. And I think to really handle these sorts of challenges well, we need to maintain the integrity of science. We need people to have a value for what science has to say. And I think, again, where we can do that, Christianity offers a great worldview for accomplishing those two important goals. And so, there's a whole lot of ways this conversation could go south and people could misunderstand. But really, my interest is in how do we care for people well, people who've been marginalized or disincentivized, because that's happened.
Yes.
But how do we also maintain the truth? Because if we sacrifice one or the other, we're missing what can be.
Right. Well, good. I appreciate the courage to tackle that question. If we don't have these courageous conversations, the problem is never going to be truly solved. Right. And so I appreciate your willingness to risk that. So,
and I appreciate your stepping out a little bit and allowing it to happen because, I don't know, we could get blowback like I said, there's all different ways that people could either misunderstand or we could have said something wrong and actually been incorrect here. So,
well, if people are watching this, on our YouTube channel or on the Reasons to Believe Facebook page, hey, if you got comments or thoughts, please share them. We want to hear your perspective on these questions, whether you agree with us or not. We want to hear your thoughts and your input. Anything else?
No,
Jeff?
let's go ahead. Transition.
Okay, so we'll go from a really heavy topic to something a little bit more fluffy and light. A Twinkie.
That is fluffy. I don't know that it's light, but it's definitely fluffy. So.
Yeah, well, and the reason I show the Twinkie here is I'm just going to have a little bit of fun for a few minutes to introduce what I want to talk about with regard to the latest advance in the sequencing of the human genome. And the point is that there's a lot of things that we think are true that actually are not.
Okay.
Right. And that we think things are one way, but in fact they're something different. So I don't know if you've ever heard that Twinkies will survive on the shelf for like a couple of decades, right? You want to make sure your bomb shelter is well stocked with Twinkies. Well, fact of the matter is the shelf life of a Twinkie is only 45 days.
Really?
So it's
that
not,
surprises me.
It's not a couple of decades. Here's another one that's fun. These are birds eating rice. And, I don't know if people threw rice at you and Lisa during your wedding when you were leaving the church.
Yeah, we changed over to birdseed because rice was bad for the birds.
Right. Well, I think we actually had people throw rice at us. But there is this idea that if birds eat rice seed, it'll swell up in them and they'll die. Well, that's not true.
Okay.
Yeah, but it's still probably great to throw bird seed. But and then this is a picture of someone who I've long respected, George Washington Carver. And the claim is that he invented peanut butter. Well, actually, he didn't.
He didn't?
No, but he did actually come up with a long list of uses for peanuts.
Gotcha. Okay.
That helped the agricultural economy in the South.
There is one of the buildings at Iowa State where I went to school was named for George Washington Carver. And I don't know that it's because he invented peanut butter, but it was because of the latter contributions you were describing.
So great American. And then, of course, here's spinach, a can of spinach. And spinach actually doesn't have an exorbitantly high iron content to it.
Oh, really?
Yeah. It's rich in vitamin A, but not necessarily overly rich in iron.
Okay.
And so Popeye didn't get his strength from the iron in the spinach, but it was from the vitamin A.
I can't trust my Saturday morning cartoons for good information.
No, I'm sorry you can't. And then last but not least, fortune cookies are not a Chinese invention.
Okay.
They're a Japanese invention. And so Japanese invented fortune cookies. And in fact, in China, people view fortune cookies as an American phenomenon. So anyway,
that's funny.
Okay, so all that tomfoolery to lead us to this slide, which is the human genome, or to be emblematic, of the human genome. And the claim is that in 2000, the Human Genome Project, in collaboration with Celera Genomics, sequenced the entire human genome.
Okay.
But that's actually not the case.
All right.
They didn't sequence the entire human genome in 2000. They only sequenced 92% of it. There was 8% of the human genome that remained unsequenced because those sequences were considered to be intractable sequences because of their highly repetitive nature. And
So what makes them untractable? Because I mean, I'm gathering for most of the genetic code, it's just laid down kind of random, not random, but distinguishable format. So it's just that it repeats a lot, makes it impossible to sequence.
Yes,
really.
There are regions of the. And let me just quickly do a background slide, and I'll take on your question, which is a great question. This is just simply showing the fact that in humans there are 23 chromosomes, and each chromosome occurs as a pair, where one set comes from the mother, one set comes from the father.
Are the sizes of the chromosomes to scale?
Yes.
Wow,
and so they're actually numbered from largest to smallest.
Okay.
So 1 through 22 are the autosomes, and then there are the two sex chromosomes, the X and the Y.
Right.
And what's shown here is not only the pairs, but then each of the chromosomes has actually undergone duplication.
Okay.
So it's showing kind of the duplicated chromosome. But here's another cartoon of a chromosome, and compared to an actual photographic image of a chromosome. And so the sister chromatids means that this is a chromosome that was replicated.
Okay.
And you notice that it's kind of roughly an X shape, where the center of the chromosome is called a centromere. This is the point of attachment for the mitotic spindle, so that when mitosis takes place, those two chromosomes are separated and pulled to the opposite end of the cell pole, so that when the cell divides, each daughter cell will have the right number of chromosomes.
Gotcha.
And then there are telomeres, which are the tip ends. And then there's an area called euchromatin, and then heterochromatin, which are, again, the telomeres and the centromeres. The heterochromatin is made up of highly repetitive DNA.
Okay.
Whereas the euchromatin is made up of DNA that isn't highly repetitive.
Okay.
And when it comes to sequencing genomes using the techniques that were available in 2000, the methods were only able to really handle non-repetitive DNA sequences.
Okay.
Because what happens is those sequencing methods could only sequence pieces of DNA that were about 200 base pairs or 200 genetic letters in length.
Right.
And so what you would do is you would take a piece of DNA that might be associated with, let's say, chromosome 1, and you would chop it up into small pieces, and you would sequence those pieces, and then you would look for places where those sequences would overlap, and then you would reassemble the sequence. Well, that works well for non-repetitive sequences, but if you're looking at repetitive sequences, it's very difficult to assemble.
Right.
You can sequence them, but it's very difficult to assemble it.
Gotcha.
And so, unfortunately, the 8% of the human genome that was highly repetitive is the heterochromatin, and it's primarily the centromeric DNA as well as telomeric DNA. Now, the location of the centromere is also important, and so there's some chromosomes are called metacentric, where the centromere is approximately at the center.
Right. And
then there's submetacentric, where that centromere is slightly above or below the midpoint. And then there is something called acrocentric, where you have two short arms. And then there's telecentric, where the centromere overlaps with the telomeres. This isn't seen in the human genome. But we do see 5 chromosomes that are acrocentric. And because of the relatively short arms, and because they're near centromeres and telomeres, those acrocentric short arms were also inaccessible to the sequencing methods as well.
So that 8% includes the heterochromatin and the telomeres because they're highly repetitive, and just because you don't have enough uninterrupted space where you're away from the repetitive stuff, these centromere or the acrocentric.
Right.
Okay, gotcha.
And there's actually a fair amount of repetitive DNA in the short arms of acrocentric chromosomes as well.
Okay.
But in recent years, there have been some significant advances in DNA sequencing technology, and I need to refer to my notes here. There are 2 techniques that are now being used. One's called the Oxford nanopore method, and this method can actually sequence contiguously about 1 million base pairs.
Okay.
But it gives you moderate accuracy. But that allows you now to sequence regions that are highly repetitive because you're not having to look at kind of reassembling, overlapping sequences, which is very difficult to do, again, with repetitive DNA sequences. The other technique is called the Poor Bio HF DNA sequencing method that gives you highly accurate sequences for about 20,000 base pairs. So by combining these two methods, in 2019, a research team sequenced from telomere to telomere the X chromosome.
Okay.
And that then led to the formation of what's called the Telomere-to-Telomere Consortium, or the T2T Consortium, that on April 1st of 2022 of this year, no joke, couldn't resist, published a series of landmark papers where they report on the complete sequence of the human genome, where they actually have now been able to sequence not quite all of that DNA that has been historically intractable, but nearly all of it. There was about 1,000 gaps in the sequence in 2000, and those gaps persisted for nearly a couple of decades. And they now have been able to close all of those gaps except for 5, which amounts to about 10 million base pairs. And
which is like 3 billion or something. So that's a pretty small fraction.
It's a pretty small fraction. Right. So this is a huge breakthrough. And of those approximately 2 million sequences, they were able to get 10 million base pairs that corresponded to centromeres, about 16 million base pairs that corresponded to telomeres, and then 67 million base pairs that corresponded to the 5 short arms.
Okay.
Now, something else that they also did to make the sequencing possible is they actually worked with a particular cell line called the CMH 13 cell line. And this is a cell line that was derived from what's called a molar pregnancy. There are times where you'll have an egg cell that doesn't have any genetic material in it at all. And when that happens, it can still be fertilized by a sperm cell, which would bring in 23 chromosomes,
right?
22 autosomes, and then the sex chromosome, and it can duplicate. It can become duplicated, and so you wind up with a cell, an egg cell that has 46 chromosomes, but all of them come from the sperm cell.
Okay.
And so what this means is that instead of having a genome that consists of 23 pairs of chromosomes, where one set comes from the mother and one set comes from the father, where the parental and the maternal chromosomes have different sequences, you now have chromosome pairs that have the same sequence. So that actually simplifies trying to sequence highly repetitive sequences, because you're not having to deal with not only the repetitive sequence, but the diversity within those sequences. And they use the cell line.
Out of curiosity, does this lead to productive offspring?
No.
Okay.
If a molar pregnancy has to be terminated, it's not a good thing.
Okay.
In this particular cell line, it has 2 X chromosomes. And they then use the Y chromosome from a male donor to fill in the Y sequence.
Okay.
And so the bottom line is that we now have really, as close as we have ever had, a complete sequence of the human genome, which is really very exciting.
Right.
Very interesting, the experimental approach, the technical approach that this team took to producing that sequence, and so this is now giving us insight into, parts of the human genome that we, for nearly 2 decades, had no real understanding whatsoever. Now, what's interesting to me is the excitement that the research community has, because we now have access to these regions of the genome. Because when the genome was originally produced, the sequence was originally produced for the human genome, people acknowledged that, again, 8% wasn't sequenced, but nobody really talked about it much, which is why most people weren't under the impression that we, had an incomplete human genome sequence. And the reason why most people didn't talk about it is most people thought these highly repetitive sequences in the human genome were simply non-informative.
Right.
That they were essentially either telomeric or centromeric DNA that were really structural features that didn't really have any kind of functional role beyond that. There were, what are called segmental duplications and satellite DNA, even in the euchromatin that people couldn't really sequence. But again, they thought this is just junk DNA. There's really no reason to be worried that we don't have these sequences. But over time, we've come to realize that these sequences are actually giving us something very important in terms of understanding the biology of the human genome, understanding the mechanisms of gene regulation, understanding genetic diversity from person to person around the world. We are recognizing there's certain diseases that have genome associations with these highly repetitive sequences that we didn't have any way to really characterize apart from now having this sequence available to us.
So that's fascinating. So it seems to me that if a problem in this highly repetitive DNA can cause diseases, or at least contribute to diseases, that says that something's going on with that. I mean, if it were just strictly junk and just off there baggage is being carried around, it doesn't matter what the baggage looks like. The fact that it actually can cause pathology is a very strong indication that it's doing something.
Yes, and what's interesting is, the kind of the master paper that kind of described the work was then followed up in the same issue of Science with 5 additional papers. And there's other papers published in other journals that are part of the T2T consortium release of data. But as they did preliminary characterization now of these, historically intractable regions, they were seeing a lot of results that indicated that this is really very important. These are really very important regions of the genome that, again, that we were missing out on and to our detriment.
Right.
That 8% gap in sequence data had real significant implications. And so one of the researchers involved in the T2T consortium, Karen Miga, said, these parts of the human genome that we haven't been able to study for 20 plus years are important to our understanding of how the genome works, genetic diseases, and human diversity and evolution. David Hostler, another member of the T2T consortium, said, we've gotten an enormous understanding of human biology and disease from having roughly 90% of the human genome, but there were many important aspects that lay hidden out of view of science because we did not have the technology to read those portions of the genome. Now, again, what's striking to me is the change in perspective that has taken place in the last couple of decades from, is this really important to have the sequence data? Maybe we want to have the data for the sake of completeness, to now recognizing that we were missing some very important information about the human genome by not having that sequence. And to me, this is reflecting the results of 20 years or so of work on the human genome, where we recognized that instead of roughly 3 to 5% of the genome being functional, the rest being junk, that we now realize that probably most of the human genome has functional significance, has structural significance that really impacts human biology, genetic diversity, and also disease and our understanding of disease processes. And so it's really highlighting the fact that we had a very naive view of the human genome in 2000 when it was sequenced, and we now are recognizing that we're in our infancy in understanding the human genome, and that to go from an infancy stage to begin to matriculate to a more sophisticated understanding requires having these missing sequence data, and it really highlights, I think, the sheer complexity of the human genome and probably the sophistication of the human genome that we failed to appreciate in the very early days of having that sequence data. And then also thanks to projects like ENCODE and the work of individual researchers independently characterizing the genome, we're recognizing that what we thought to be unimportant, junk DNA sequences is actually turning out to be turning out to be important. And so this is kind of continuing
Right.
in that mindset. That mindset is being, further elaborated on that, hey, what we thought was junk really is very critical.
Well, that's what struck me as I was listening to your discussion. I know we've had, tried to. I've talked with you about the ENCODE project just to kind of understand what it is and the implications of it. And remember you describing this kind of argument going on in the scientific literature and the community about, is the ENCODE really saying that 80% is, or is it less than? And, there's, evolution says this, so therefore that can't, there's all sorts of this stuff that's going on. But the ENCODE data really did, it seemed to be indicating that it was all pretty important because all of it was doing something.
Right.
The fact that we've gone in and in that 8%, it's not like, oh, there's a couple more things. It's like, oh, there's a whole bunch more in here. Seems to validate that what the ENCODE was saying is that, this is actually important stuff. So it kind of weighs in favor of the entire genome being useful rather than bits and pieces and most of it being junk.
Yeah, and of course where this plays out from an apologetic standpoint is that if we take the view that human beings are the product of God's handiwork, that life on earth is the product of God's handiwork, of course it raises the question, well, why on earth would God create genomes of organisms that are littered with non-functional DNA, right? And then why would these non-functional DNA sequences be shared, among organisms, right? That seems more like what you'd expect from an evolutionary origin. Now, but, so from somebody that advocates for a creation model perspective, an intelligent design perspective, the first thing that we have to do is show that, hey, this is actually functional. Because if it is functional, then it at least says that it's legitimate to think of the genome as the product of God's handiwork.
Right. Yeah.
And that maybe these shared sequences are reflecting a shared design as opposed to common ancestry. So, this is actually supports our creation model approach. It doesn't necessarily undermine someone who would view the genome in evolutionary terms. Although, part of that debate that is going on is interesting because on one hand, you have people that are working in genomics, like the two researchers, David Hostler and Karen Miga that we quoted, who see the information from ENCODE and from these genomic studies as being incredibly valuable.
Right.
And then you have evolutionary biologists, on the other hand, who are decrying the idea that most of the genome is important, that most of the genome is functional, because it actually undermines things like the resolution to the C-value paradox. It undermines ideas that are endemic in molecular evolution.
Right.
And so I remember debating P.Z. Myers, and a biologist and an atheist, and the issue of the ENCODE project came up during our exchange. And he said the ENCODE project results can't be correct, because if they're correct, then evolution can't be valid. Right. And I thought that was very interesting, because it's, I think that sentiment connects to the point you were making, is that science is done in a particular way, that it's data that evaluates theories. Theories don't evaluate data, right? And so this is an example of, again, kind of a bizarro world approach to science, where suddenly the data and the work by researchers producing the data is being evaluated in light of a theoretical expectation, as opposed to the other way around.
Right.
And so, whether, again, one views humanity's origin in evolutionary terms or from an ID creation model perspective, that statement of P.Z. Myers is deeply concerning.
Right.
Because it's undermining the nature of the scientific process.
Well, and, I'm not going to elaborate too much, but that's the sort of stuff. There's enough things that come up where conflict about the ideas makes it hard to get to the truth. How that plays into, if you now bring racial discussions into that could make that conversation just that much harder.
Right.
Things could be perceived as racial when it really is just scientists have really strong ideas about stuff. But I think a bigger point is, this strikes me as a way for a budding scientist or someone who's looking, how can I make my mark, or how can I do something? It's those places that come up where, wow, this seems redundant, or wow, that looks irrelevant. if you have that's probably there for a purpose. A lot of people are going to say, oh, that's not important. You're going to have some unfettered time to work on something, and you're probably going to discover something because historically things that we've thought were unimportant or junk turned out to be more important than we thought they were. It actually can direct you to the ideas that are going to be productive and fruitful by thinking of it in terms of that design or creation perspective. So.
All right, well, that's all I've got, Jeff. I appreciate your contribution to today's show. I'm going to go ahead and wrap everything up, and just want to say thank you to all of you who watched the program or listened to the podcast of this episode of Stars, Cells, and God. I hope you found the conversation helpful and encouraging and provocative. Please, again, join the discussion by offering your thoughts in the comment section. If you also found this program helpful and insightful and interesting, please share it with other people through your social media or through other means. And also remember to like the video and to subscribe to more content on our Reasons to Believe YouTube channel. And new episodes of Stars, Cells, and God are dropped every Thursday. And also, if you go to our YouTube site, there's all kinds of other programming that I think you'll find interesting. Also, make sure you follow us on social media, RTB_official, to learn more about what's happening here at Reasons to Believe. And thanks again for watching. And remember that the more we discover about science, the more we have reasons to believe.